Hello, students of Cesar Chavez High School. My name is Gabriela Espinosa, and I'm a molecular cell biology major at Cal State University Long Beach. And today I'm going to be talking to you about viruses, although they can be very scary, so be prepared. So you may ask, what is a virus? Well, viruses are non-living infectious particles. They are obligate intracellular parasites that can only survive by invading a host cell. For example, these hosts are humans, plants, and animals. So yes, don't think that not only us humans are susceptible to being infected by viruses, so are plants and animals as well. Here on the slide, we have a very beautiful picture of the Ebola virus. So although a lot of viruses may not look what is pictured on the slide, like the Ebola virus, we're able to see and kind of get a good picture of what can we expect of how these viruses should look like. So next, after learning about what is a virus, we're going to talk about the job description for viruses. And they have these three main goals, three main steps of what their job entails. So the first step and or purpose is to invade and conquer the host immune system. Second of all, it is to initiate replication, multiply in huge numbers, and infect surrounding cells. And last but not least is to stay alive. Although these viruses are non-living infectious particles, they are dependent on the longevity and health of the host. So if the host dies, no more replication, but if the host is alive, perfect enough to continue replication and continue spreading that infectious disease inside the host, and as well as giving it a chance to infect other individuals who are healthy and are capable of being infected by this virus, or by any virus in any matter. Here we have a quick picture of how we can see how a virus invades our immune system and basically renders us to be very weak. For example, we have the HIV virus. It is the human immunodeficiency virus. So what we see here is that once a virus particle enters our systems, it is going to attach using the virus cell receptors to the targeted healthy cell that we have in our bodies. Once that receptor makes contact with the lipid membrane, it is entered into the host cytoplasm and it begins reverse transcriptase in order to make tons of usable viral DNA copies. Once that happens, those viral copies will insert itself into the nucleus, causing there to be a huge genome replication of that infectious virus particle. For instance, we can see here that the HIV RNA transcript will be used to make tons of viral infectious proteins that will be assembled with the respective genetic content and capsid so that they may be released outside of the host cell and continue to spread the infection around the host. So now that we've talked about what a virus is and everything and how it does its job, we got to examine the virus in its entirety of the blueprint. So how can we classify viruses? Well, we can classify viruses by looking at the infected particle itself, the virion, and the capsid. I've stated before that the capsid is the protein coat. We have to look at the shape and symmetry of the capsid, genetic composition, whether it may have DNA or RNA, and see if it has an available or absent lipid membrane. Here on the right-hand screen, we can see a really nice picture of 7 different types of viral genomes. Here we have double-stranded DNA, single-stranded RNA viruses, positive and negative sense, and their respective orders. And basically, in order for these viruses to be transcribed, they all have to have a really nice completed piece of mRNA. mRNA is responsible for encoding these viral proteins and allowing the virus to be spread out through the entire body. In terms of shape and symmetry of the capsid, we see here on the lower left that there is a picture of the HIV capsid. Notice that it's not a standard circular shape that we're used to seeing. It is very curvy in a shape, in the sense of a potato, if you want to think about it. And not only is every virus looking like a small potato, think about it that they take on shapes like the curly fries from Jack in the Box or a straight rod or pole. And so that's what makes a lot of these viruses very, very different from each other, how I have stated in the beginning of this video. So yes, although viruses are super duper scary, we must remember that these viruses are the frenemy of our world. They have the potential to cause catastrophic events in our livelihoods and our surrounding environment. And so remember the measles outbreak that happened many, many years ago? It was infecting a lot of our youth. And so through a lot of countless of research and trials, we were able to ensure, maintain, and preserve the public health of everyone by creating a vaccine. And so since we've created that vaccine, we now use that to our advantage. So every time we have to go get our measles vaccine, we have to basically walk to the doctor's office and say, can we get the vaccine? And that prevents us from coming into contact of the virus itself. So since we have that vaccine, we can no longer get infected of that virus. Cool, right? And so thank you for coming to my presentation of viruses. And I hope that this presentation has inspired you to go out there in the world and go discover viruses for yourself. Hopefully you will all one day want to take into consideration of being a biologist and seeing how these viruses are truly beautiful and scary at the same time. Thank you.